So, well, first, let's talk about like what is a neurotransmitter? What are the different types of neurotransmitters? So, we effectively going to be talking about two types. One type is the small molecule neurotransmitters. These molecules act fast, but the responses that they cause do not last long. So I'm going to put the note that they are short-lived responses. Now, obvious question that you should ask me, do we need to know all of that that's in the table? And the answer is no. Your life's going to be much easier. I'm going to explain right now what you absolutely must know for the exam. So first of all, you need to know for 7 neurotransmitters. I am highlighting them right now. I think 7 or 8, all the way up to glycine. Only this, 8 neurotransmitters. The only thing you need to know about them is whether they are excitatory, inhibitory, or both. Am I making sense? Yes? Okay. So I can ask you a question like, which of the following neurotransmitters is excitatory? And you pick like exclusively excitatory. Are we good right now with that idea? Okay, excellent. Now, a common question that I sometimes face, like acetylcholine, which one is that? It's either, it's both. Okay, it's excitatory in certain parts of the nervous system, but in general, it can exert both effects. So far, we're doing all right? There will be absolutely no questions on the site of action. I 100% promise you that. And you don't need to memorize the associated drugs. But this last column will be very useful when you're going to prepare yourself for the questions about agonists and antagonists. And this is something I'm going to be talking about in a very short time. Okay? So again, small molecules act fast, responses do not last long. Please know whether they are excitatory or inhibitory or both. And the last neurotransmitter that you need to cover is glycine. There will be no ATP, adenosine, nitric oxide, or endocannabinoids on the exam. Next idea is neuropeptides. So neuropeptides are large. They act slowly, but their effects last long. So I there are only 2 categories of neuropeptides that I want you to know, to remember. Inhibitory endorphins and enkephalins, and excitatory tachykinins, such as substance P. These neuropeptides are truly neurotransmitters that act in your brain. For example, endorphins and enkephalins work as natural opioids. They inhibit pain. They're produced when you are excited, right? And for instance, when you exercise. And they provide this inhibitory effect if you are hurting, okay? Kind of explains some of the ideas that I will introduce to you in a minute. So, do you need to know anything like this part? No. I'm not going to ask questions about that. But what I am going to ask is the idea of agonists and antagonists. Okay? So let's talk about it. What is, I don't know, what the hell? No, seriously, hold on. It's very annoying. For real? Okay. I'm going to use some space over here, and I'm going to use the data in that last column to make examples for my talking points. So what are agonist versus antagonist? Remember, we're talking about the fact that neurotransmitters act on receptors, right? So imagine we have a receptor and we have a neurotransmitter that can bind to it. Okay? And that results in some kind of signal. Well, if you start employing agonist, and call it AG, it will also bind to the receptor and will also produce the signal. And maybe the signal will be even stronger. Okay? Well, good. I'm going to give you some examples, all right? So, in case of antagonist, you do have a receptor, okay? And you have a neurotransmitter that can bind to it. But when you use an antagonist, it will bind to the receptor, block neurotransmitter from binding to it, and there will be no signal. Do you see the difference in the relationships between the agonist and antagonist? The most important thing is that both of these types of molecules, they bind to the receptor, the target receptor. So let's take a look at some examples that we have here. So a really good example of the agonist is nicotine. We have receptors for neurotransmitter acetylcholine in the brain, and nicotine binds to those receptors, and it binds much better than acetylcholine. So it provides a pretty strong stimulation of those receptors. And if you ever, I'm not judging, I'm not asking, if you ever tried nicotine products, you know, they're delicious. I smoked for 22 years. I still, every day, I quit 7 years ago. Almost 8. Every day I miss smoking because it was great. It was really bad for my health, but on the psychological level, it's fantastic. I'm not endorsing this, by the way. My point is, this is an example of agonistic action. Okay? Am I clear? Another example of an agonist are various synthetic opioids. They are drugs that treat pain. They act on the receptors to which your normal natural opioids, such as endorphins, bind. The problem with things like heroin or morphine or OxyContin is that when they bind to those receptors, they bind way better and produce much stronger signal, which is great if you treat pain, but the problem is that the receptor eventually becomes resistant to that stimulation. That's agonist. That's all agonists. Okay? Now, an example of antagonist, although it's not going to be in the central nervous system, have you heard about the drugs called beta blockers that I used to treat hypertension? They are antagonists of beta-adrenergic receptors found in the heart. They reduce the force of contraction and frequency of cardiac contractions, therefore, reducing blood pressure. Okay? Before we move on, does that make sense? The idea of agonists and antagonists. Excellent. Because a lot of drugs, whether we're talking about pharmacological interventions or we're talking about, drugs that, illegal drugs like, they act as either agonists or antagonists of certain receptors. Now, why this is important? So, last week when we were talking about, synaptic transmission, we mentioned that neurotransmitter has to be destroyed at the end, right? And a lot of drugs, they mess up this destruction. So here are some examples. For instance, antidepressants and cocaine block reuptake of norepinephrine. Norepinephrine is effectively a feel-good neurotransmitter. This is why antidepressants work, and this is why people on cocaine are so happy and energetic. Because norepinephrine, instead of being taken into a presynaptic neuron, stays in the cleft. Okay? Another example of cocaine messing around is cocaine blocks the reuptake of a neurotransmitter dopamine, which is also a feel-good neurotransmitter. Now, you're probably familiar with the drug called Prozac. Prozac is one of the inhibitors of the serotonin reuptake. Serotonin is an inhibitory neurotransmitter which makes you calmer. So, when people take antidepressants, it makes them calmer. It reduces their anxiety. Okay? And if you think about this, for instance, here's the deal. So here's an example of antagonist. There is a neurotransmitter called glycine. Glycine is an inhibitory neurotransmitter in the spinal cord. Your spinal cord transmits signals to your muscles. Follow my train of thought, okay? If there is an excitation signal that is sent to your muscles, what do you think your muscles are going to do? They're going to contract, yes. If there is an inhibitory signal, what do you think they're going to do? They're going to relax. Yes. It has two steps. I'm not going to go into the weeds, but you understand the idea. If your brain sends excitatory signal, muscle contraction. Inhibitory signal, specifically glycine, relaxation. Okay? I don't know, anyone heard about this drug, strychnine? Strychnine was widely used in 19th century and early 20th century as a rat poison. Strychnine is an antagonist of glycine receptors. If strychnine binds to those receptors, glycine becomes useless and there is no more inhibition. Does that make sense? If your brain cannot deliver inhibitory signals to your muscles, what happens to your muscles? They all what? They all contract, which is not great because you want your muscles to contract and then relax. One of the most important muscles to relax is your diaphragm. If you don't relax it, you can't exhale. Strychnine causes respiratory arrest. It was pretty widely used by 1/2 of human beings for taking care of another half of human beings. Any ideas who 1/2 was? Well, obviously, strychnine is a poison, and if you use it on a human will die. We're talking about women. Because in the 19th century, if a woman wanted to get rid of an abusive husband, or get rid of a husband, this is not a joke. It was very, very common. Because women could not physically affect a more stronger man. So they would just add it to the food. The problem with strychnine, it can be sensed in the food, so the food has to be spicy. And it's been a known evidence if for some reason some British woman cooks some super spicy food, which is very uncharacteristic of British cuisine, probably this dude had it coming. Okay? Now, what type of a question you can expect regarding agonists and antagonists? Let's say this. Did we talk about the sludge syndrome, no? Okay, so I'm going to sort of capitalize on this. So I'm going to talk about neurotransmitter called acetylcholine. A category of drugs called organophosphates, which initially were used as pesticides. Organophosphates inhibit the degradation of neurotransmitter. Okay? So I've got a question for you. If we block a degradation of neurotransmitter, what's going to happen to the strength of signal that goes through the synaptic cleft, huh? I can't speak louder. It's gonna neurotransmitter is gonna remain in the synaptic cleft. Do you agree with me? If it remains, does it keep signaling? Yeah. Imagine that. So remember my analogy with a doorbell. Normally, you press on a doorbell and remove your finger. That removal of the finger is the inhibition, removal of neurotransmitter. If you can't remove, your finger is stuck to the doorbell, so you keep ringing. Okay? And the signal that acetylcholine produces, the commands that it gives, results in this: salivation, lacrimation, urination, defecation, gastrointestinal cramps, and E stands for emesis, which is vomiting, which is not great when it happens all at the same time. So organophosphates were later converted into chemical weapons. Gases like VX, Sozoma, and Ozarin are all organophosphates. Just a little side note, chemists of which country do you think converted them to chemical weapons in about 1930s? Yes, Germany, of course. It was Germany. Now imagine there's a soldier on the field, and I'm going to give you a little hint. I'm going to make a drawing here. Okay, so this is your presynaptic neuron, right? This is your postsynaptic neuron. These are your neurotransmitters. And remember, they are not destroyed. So here's my question. A neurotransmitter, what does it have to bind to? Can you say it louder? Receptor. Thank you. So these are receptors for this neurotransmitter. Remember, person is poisoned. Neurotransmitter isn't going anywhere. You need to not let that person die. What type of drug? I'm not asking the name of the drug. What type of drug would you give that person? We just talked about it. The answer actually is right on the screen. Say again? Antagonist. Yes, an antagonist. So you want an antagonist to be given to block the receptors. Clear? And the name of that antagonist in this particular case, atropine. You may have heard the name of the drug. It's used in pharmacology for a variety of reasons. Okay? Does that make sense? So the question that you can expect is, you have a patient with organophosphate poisoning, and don't worry, you don't need to memorize what happens in organophosphate poisoning, because I'm going to tell you. I'm going to tell you that organophosphates block the degradation of acetylcholine. How should you treat that poisoning? And you pick an answer. We should give a patient an antagonist for acetylcholine receptor. Does that make sense to you all? Okay? Good? Right. So now let's move on to the conversation about action potentials. And as you already, I guess, figured out, I'm not particularly keen on you memorizing numbers. So you don't need to memorize any numbers from that graph. I want you to know the relative concentrations of 4 ions: sodium, potassium, calcium, and chloride. And by relative concentrations, I mean this. The concentrations of sodium, calcium, and chloride are higher outside of the cell. I think we mentioned that before. The concentration of potassium is higher on the inside. There will be absolutely no questions about any other ion except these 4. Okay? So now let's move on to the idea of ion channels. We had a conversation about, the diffusion. Remember, ions cannot diffuse directly across the membrane because they are polar, they are charged. They have to use the channels. There are 4 types of channels that you must know. You must know how they open and close, and believe me, it's nothing super complicated. So the first type of channel is so-called a leaky channel. Leaky channel randomly opens and closes, allowing ions to diffuse across the membrane. Quick question: what will determine the direction in which a particular ion diffuses? Gradient of concentrations, exactly. If this leaky channel is for potassium, potassium will diffuse out of the cell. If it's for sodium, it will diffuse into the cell. Then we have voltage-gated channel. What opens and closes voltage-gated channel? Electricity. Electricity is the movement of charged particles. Whenever there is a change in the charge on the membrane, a voltage-gated channel can open. If the channel opens in response to a neurotransmitter that binds to the channel, we call such channel ligand-gated or chemically gated, usually ligand-gated channel. Sounds good? So neurotransmitter can bind to a channel, we call them ionotropic receptor, and open it, and the ion will diffuse in whatever direction the gradient makes it. Okay, good? And then mechanically gated channels. Mechanically gated channels open and close in response to mechanical stimulation of the membrane, like a deformation, like pressure, vibration, stuff like that. Are we doing all right so far? Okay, so please understand what stimulus opens each of these four types of channels. Before, go on.
So for the it's when it's presented with a neurotransmitter,
Chemically gated or ligand gated channel will only open if a correct neurotransmitter will bind to its receptor part. Okay? Does that make sense? Like, I always give analogies, I know. I have a fob that opens doors at Lakeland. And I have a card that opens a door at Tri-C. In order to get here, I need my Tri-C card, not my Lakeland card, because it's the proper card. So the channel that is gated by acetylcholine will respond to acetylcholine. Okay, good? Now, the worst slide I've made in my life. So what we're going to do, we're going to completely ignore not the content of the slide, but the way it is written. And I'm going to explain it to you. Specifically, I'm going to explain you the concept of Nernst potential, which is quite an important concept. So let's say this is the membrane. You see the membrane. This is outside. And this is the inside of the cell. Now we're going to start with potassium. Just imagine, I'm not going to draw any channels, but imagine that the only channels that exist in the membrane are potassium channels. So potassium concentration is higher on which side? On the inside. So potassium will diffuse through those channels to the outside of the cell. Agreed? What type of charge will it bring to the outside of the cell? Positive. Potassium is positively charged. What type of charge it will leave behind? Negative. Would you agree that positively charged potassium would be drawn to negatively charged interior of the cell? Okay? So the outside movement is dictated by the diffusion. Okay? The movement of potassium back inside is due to the electrical forces. Do you find it agreeable that at some point, at some distribution of charges, at some negative charge on the inside and outside, the flow of potassium outward will be compensated by the flow of potassium inward? Yeah. So the cellular potential at which flow of an ion is zero is called a Nernst potential. Okay? Yes. So the potential on the membrane, the distribution of charges on the membrane, at which the flow of an ion in and out of the cell are equal. Flows cancel each other out. That specific potential is called a Nernst potential. It is a theoretical value. You can calculate it, but in nature, it doesn't exist. Okay? Now, the potential is always measured inside the cell. So, the Nernst potential for potassium, will it be positive or negative? Negative. It's inside. Now, let's figure out sodium. The concentration of sodium is higher outside. So it will diffuse in, bringing what charges into the cell and leaving behind. So electrical forces will drive sodium back, and the Nernst potential for sodium will be positive or negative? Dude, positive, because it's inside the cell, okay? Now, if you look at the numbers, which I can't emphasize it enough, you don't have to memorize. The Nernst potential for potassium is negative 94 millivolts. The Nernst potential for sodium is plus 61. Again, whatever numbers are. Now, I'm going to tell you something, that the actual distribution of charges, the actual membrane potential, is defined by these two ions, and that's it, sodium and potassium. And if I would ask you, what would you do? How would you find the actual membrane potential in a cell? What would you do with these numbers? It's like -1 out of 10 question. It's sodium and potassium, only two ions. One gives you -94, another gives you plus 61. What would you do with these numbers if I would tell you only two ions form it? So again, add them. Yes. In reality, potassium brings more. Its impact is greater. This is why resting membrane potential of a cell is usually negative 70. Do you need to memorize that number? No. I'm just telling you. Okay? So RMP, or resting membrane potential, is an actual value which can be measured, and yeah, you can do it. You can measure it. Okay? Quick question. What if you have some hypothetical ion and its concentration outside is equal to its concentration inside the cell? What would be its Nernst potential? Yes, because there is no net flow to begin with. Okay? All good? Okay. So the last detail about resting membrane potential, if you look at this image right here, you can see fairly clearly that sodium and potassium are two ions that determine the resting membrane potential. But look at this. If you let these ions constantly leak, eventually concentrations may equilibrate on both sides. So in order to maintain the concentrations at the same level, there is a sodium-potassium pump. If you look at the movement of ions, sodium diffuses into the cell, but being pumped out of the cell. Potassium diffuses out of the cell, but being pumped back into the cell. Now, I want you to understand that the situation that we're looking at, you may think of it as like, some fundamental law of nature, like it was observed, and then all those channels and pumps were discovered, and when they were discovered, that explained how membrane potential works. Am I clear? Now, what if you open some voltage channel? Okay. This is very annoying. Okay. What if you are opening the voltage? Not the voltage, sorry. What if you open sodium channels? Just sodium channel. Sodium will diffuse where? So influx of, say, sodium, bringing positive charges in the cell. Agreed? Which makes the cell I would say less negative. Because it's not positive yet. You see, it's still negative, but less negative. So we call this process depolarization. What you see here is depolarization. Now, when cell becomes depolarized, when cell becomes less negative, it is excitation. Clear? So these things, they go hand in hand. Bringing in positive charges depolarizes the cell, and cell becomes excited. What if you open potassium channels? Where would potassium go? If you open potassium channels, where would it go? Outside. Taking positive charges out, making cell more negative. Alternatively, you can open chloride channels. That will bring negative charges in. This situation is called hyperpolarization. So, more negative equals, I don't know what I'm writing, hyperpolarization, and becoming more negative equals inhibition. Okay? Well, good? So now let's take a look at this. The idea of a graded potential. So this right here is a stimulus. What type of stimulus? None of our business. It can be a neurotransmitter, it can be an electrical signal, it can be a mechanical signal. The point is that this stimulus opens sodium channels. And sodium diffuses in which direction? Inside. And cell, does it become more positive or less positive? More positive, which means, yes, it becomes depolarized. Okay, we're good? Locally depolarized. But the issue is that that stimulus acts on a very small patch of a membrane. So that depolarization, that change, will eventually dissipate. One of the classic analogies of what a graded potential is ripples on the water. Imagine that you throw a boulder in the water. There will be ripples. What's going to happen with them as they move away from the place the boulder touched the water? They get smaller, right? And eventually they disappear. Same happens to the graded potential. The electricity essentially leaks out. Right? Make sense? One of the reasons why we call this a graded potential is because it can be bigger or smaller. Let me give a very, very crude but simple example. So let's say that's your resting membrane potential, and you open one channel. Okay? So the cell becomes more positive and then goes away. Well, good? Now you're going to open 10 channels. So more ions will diffuse. Do you agree with me? And that change, that graded potential, will have a different value. That's really important. Graded potential can have different values. It's not yet a signal, because if the signal disappears, What's the point? But turns out that the signal can turn into an action potential. There are a lot to unpack in that image. So I'm going to start with, guess what? An analogy. Imagine that you're all familiar with the domino principle, right? Yeah? So imagine you have dominoes set for demonstration of a domino principle. These are unusual dominoes, though. Each piece is like 10 feet tall, weighs 1,000 pounds. So they're really heavy, they're really big. And they're not that easy to tip over. So you approach the very first one and give it a push. And it vibrates a little. It doesn't fall, though. Okay? You give it a stronger push, it vibrates more, still doesn't fall. So those different vibrations, because you gave it a different strength of a push, are examples of graded potential. Now you run and smash into that first bit. And you finally tip it over. Do you understand what I'm trying to say? Now it falls, and the next one falls, and the next one falls, and the next one falls. And eventually, that signal, that action potential, is propagated, is moving through the membrane without any additional input on your side. So, effectively, you need to cross a certain threshold. Okay? Am I making sense? So, I'm going to do schematics, and then we're going to get to this beautiful images, all right? So this is how I prefer to explain what the action potential is, and it's pretty reasonable. So imagine the green color, so green is ligand-gated channels. Red is voltage-gated channels. Okay, now, follow my thought on this. I'm going to try to fit it here. Okay. So imagine that we have a ligand-gated channel that is closed, and nearby we have a bunch of voltage-gated channels that also are closed. The membrane is at rest. Okay? So what happens is neurotransmitter binds to the ligand-gated channel and opens it. Right? So I'm going to open this channel. It's open now. All these channels are for sodium. So when this channel opens, where does sodium go? Inside or outside? Inside the cell. It makes cell more positive, right? Depolarized. Okay? Now, these red channels, how are they gated? So again, voltage. So when you have all those extra pluses on the inside of the cell, all those extra pluses open the first voltage-gated channel. It's all sodium, so now sodium diffuses into the cell, adding more pluses. Are we clear? Which opens next channel. This channel adds even more pluses, which opens the third channel. Are we clear? So you have a self-maintained opening of channels, and they keep opening, and those dominoes keep falling, And that is now an action potential because you crossed the threshold. Okay? Does that idea make sense? Okay? Now, first we're going to take a look at the events. And we're going to start with the resting state right here. Okay? So this is your resting state. These are the gates. This is sodium channel on the left, this is potassium channel on the right. In both cases, are gates closed?
Yes.
Is there any flow? No. I'm going to ask you a bunch of really stupid questions. So now, sodium channel gets open. Whether it's a neurotransmitter or electrical impulse, doesn't matter. So sodium diffuses in. Influx. Okay? And brings a bunch of positive charges into the cell. Okay? Cell becomes real positive. And when it becomes super positive, potassium channel opens and sodium channel closes. See that? Potassium starts moving out, making cell more and more negative. Good? Potassium channels are slow. Which means when the cell gets back to the resting state, they don't really close, they remain open for some time, which results in a cell losing some extra positive charges. Okay? So first, I'm going to do I have this right now? Yeah, this. I needed that. Hold on. So what I'm going to do, I'm going to step by step illustrate that image. Okay? So we're going to start with the resting state. This is your resting membrane potential. Okay? Next, we are going to open sodium channels. So when sodium channels get open, what happens to the membrane potential? Does it become more positive or more negative? More positive. So it goes up. When the membrane potential becomes more positive, is it depolarization or something else? And this happens because sodium diffuses into the cell. Okay? Now, right here, sodium channels close and potassium channels open. And potassium starts diffusing out. Okay, sounds good? So the cell goes back to its normal state. What do we call this? Repolarization. Cell repolarizes. But remember, those channels are slow. Correct?
Yes.
So potassium channels will stay open. And this overshoot is called hyperpolarization. And then eventually sodium-potassium pump brings it back to normal. So both hyperpolarization and repolarization happen due to the potassium ions get now. Okay? Make sense?
Yes.
I will absolutely ask you about this graph. You can expect, you need to know in which direction ions move. Like what happens during repolarization? Okay, like potassium channels open and potassium diffuses out of the cell, stuff like that. Okay? Now, I wanna go back to, I have no idea, this is one of the weirdest. Okay, I wanna go back to this. And I want you to specifically look at the gates. You see these gates over here? Yeah? So sodium channel, specifically sodium channel, is it open? No. This sodium channel, is it open? No. It's closed, but is it closed differently? Yeah. So this is called an activation gate. And we will figure out why this inactivation is so important. Okay? So this graph that you see here simply shows you what's going to happen if you will take an actual electrode, stick it into the cell, while the action potential is running through the axon, and measure the voltage. You're going to have that exact picture. Okay? Yeah? No? Maybe? So you measure electrical, okay. Exactly, it is. It is exactly the same. I'm just telling you that it was measured. So think of it this way. I take a rabbit, sacrifice it, take neurons out of the brain, put them on a dish, take an electrode. It can be done. I know people who do that, and stick the electrode into the axon, and then I send some kind of a stimulus, and the neuron produces an action potential. So this electrical impulse runs through the axon. And when this changes in electrical potential, when they happen right here, this is what a device, a machine, whatever, this is what it will register. Okay? Does that make sense? So we have what we call a wave of depolarization. Now, when your domino bits fall, would you agree that when it falls, it either falls or it doesn't? There's no falling, but not super strong like falling. It's just falling, okay? So this is one of the main principles in action potential transmission. It's what's called all or none. If your initial stimulus, remember the green and red circles that I used before? If your initial stimulus is strong enough to cross the threshold, there's going to be an action potential. So here, for example, initial stimulus is not enough, and these two are sufficient. But how do you distinguish between the strengths? So, in other words, you constantly distinguishing stimuli based on their strength. I can speak very loudly, I can speak very quietly. Okay? So you tell it apart not by the numerical value of action potential. It's always the same. But the frequency of action potentials that are generated. So the stronger signal will generate greater frequencies of action potentials. Does that make sense? This is called all or none principle. And if you, for instance, here's an idea. We will come back to that concept when we talk about muscles later. But right now, imagine that you are lifting something heavy, like very heavy. You're working at your limit. And your entire brain is focused on the effort. And if somebody tells you a joke, you're going to drop this. Because your brain literally sends action potentials to your muscles at the maximum frequency it can. Okay? Does that make sense? Now, why is that so important? The sodium channel, right here, isn't just closed. It's inactivated. So let me ask you a question. When I did this picture with the, sodium channel opening and then voltage gated channel opening and then another channel opening and then another channel opening, every time I do it, I show you that the signal goes only in one direction. But why? And the answer is because these channels, the previous channels, they become inhibited. They become inactivated. So when you look at this image right here, this inactivation gate, nothing can open it. There is no stimulus that can open this gate. Sodium channel remains completely closed, no matter what happens, for several milliseconds, which means the signal can only move forward. You can think of it this way. Imagine that you are running through a hallway. There are a bunch of doors. Usually these doors, the doors open both ways, right? But in that particular situation, you can only open them forward. Once they close behind you, that's it. So the only direction you can run is forward. Am I making sense? So the period of time when sodium channel is inactivated is called absolute refractory period. Nothing can generate action potential. Okay? And that explains why action potential moves in only one direction. Now, another thing that I want to ask you. So this red dashed line that I just placed here, this is a threshold for action potential to be formed. So would you agree that this is the initial depolarization you need to produce to keep action potential going? Yeah? Would you agree that the arrow 2 is longer than the arrow 1? Yes. What does that mean? It means that if you apply stimulus during that time, you can create another action potential, but the stimulus has to be stronger because you have to depolarize more. Does it make sense?
Yeah, so is that kind of why, like in that first picture you showed us up there, like the, yeah, sorry.
This one?
Or no, the one where it's like, there's multiple like bars in a row, the all or none.
Right.
Is that why, like the stimulus being stronger is what?
This, not quite. This is, well, actually, yeah, you're right. Yes. Stronger stimulus, you absolutely correct. Great note. I was thinking about something else. You are absolutely correct. Because if the stimulus is strong, it can generate an action potential right here. Correct. Yes, great. So that period is called relative refractory period. So please know the difference between the two, what determines them. Now, let's take a look at these neurons here. So this is dendrite. Dendrite receives the stimulus. Let's say it's a neurotransmitter. The stimulus generates graded potential, and eventually, just like ripples on the water, it goes away. We good? That's it. Now, what if you have enough voltage-gated ion channels that are close enough to each other to maintain that domino effect going? Does that make sense? You're going to have a proper signal transmission. But you have to place those ion channels close enough to each other, because this neuron is non-myelinated. It means that the electrical charge leaks away, dissipates away from an axon. Now we're going to provide myelination. Do you remember what myelin does? Insulates in what way? I mean, you can have thermal insulation, this electrical insulation, right? It prevents the dissipation of charges. So, these are myelin sheath layers. These are what's called node of Ranvier. So now, depolarization lasts much longer, and you only need to place sodium channels within the nodes of Ranvier, which means you don't have to open like a gazillion sodium channels, which means you can transmit the signal way faster. As a rule of thumb, myelinated neurons are fast, non-myelinated neurons are slow. We have both, and I think I mentioned the disease, multiple sclerosis, in which myelination is gone, and formerly fast neurons can't transmit the signal, and usually results in paralysis. Now, that type of transmission from one node of Ranvier to another, it's called saltatory conduction. Now, this is a really simple concept, like seriously simple. When you have synaptic transmission, usually one synapse does not generate action potential. You need the addition, okay? When neurotransmitter binds to the receptors, it can either cause depolarization. We call it excitatory postsynaptic transmission because sodium diffuses it. Or it can cause inhibition. That can happen because of chloride going in or potassium going out. And we call this inhibitory postsynaptic potential. You can imagine that a single neuron receives signals from many other neurons. And the signals can add one to another. So first I want to clear that picture right here. This picture represents a summation in which excitatory and inhibitory postsynaptic potentials cancel each other out. Am I making sense? Yeah? So no signal will be produced. Then we have this situation. You see these graded potentials. They don't reach a threshold. Although there is a stimulation, there's a neurotransmitter release, it's not enough. Can we do something about it? We can add excitatory potentials together. There are two ways. There's a temporal and spatial summation. So just to your point about summation of neurotransmitters, when you add another strong signal, on the relaxation time, they overlap. They increase depolarization, finally cross the threshold, and you have action potential, and you have a signal going through the neuron. Okay? I'm going to give you an analogy, of course. Or you can send two different excitatory signals at the same time to two different synapses, and they will add up. So here's my analogy. Imagine that your car is stuck in the snow, and you try to get it out by approaching the trunk, pushing it at one time, just pushing it, stepping away and doing nothing. I bet you won't be successful. So what you do now, you push it once, and while it's still moving forward, you push it again. So you start increasing the frequency. That's the temporal summation. Option two, you call your friends. your friends get around the car and you all push at the same time. This is the spatial summation. Okay? So different places, same time, spatial. Same place, multiple times, temporal. And this brings us to pretty much the conclusion of our conversation here. The pools and circuits. So, okay, neuronal pools, a pretty simple idea. Neuronal pool is the set of postsynaptic neurons that presynaptic neuron acts on. So think of, like, let's use us as an example. Let's say I am a presynaptic neuron. Y'all are postsynaptic neurons. I give you a signal. Okay? Clear? So the folks who are closer to me are in my discharge zone. It means that when neurotransmitter reaches them, they generate action potential. You're farther from me. When neurotransmitter reaches you, it's not enough. You are not at the threshold level yet. But what happens is you are slightly depolarized. You are facilitated. You are getting closer to the threshold. Questions? Basically understand the difference between discharge and facilitated zone. Finally, circuits. Neurons are organized. You have to understand it's a very branching system. Okay? The only organism for which we know the neuronal map, as far as I know, is a worm called Caenorhabditis elegans, and it is like 100 plus neurons, and it took scientists like 20 years to figure this shit out. So with humans, it's not going to happen ever. But circuits, different organization of neuronal signaling is used for different activities. For instance, diverging circuits mostly control muscle activities. So if you look at this circuit, a single neuron sends signals downstream to many other neurons. So signal gets amplified in a way that it's branched out. So basically one neuron in your cortex can activate multiple neuromuscular junctions in your muscle. Converging circuit is the opposite. Several neurons all converge on the same receptive postsynaptic neuron. It's very common in sensory transmission. One of the examples, your olfactory neurons that sense smell, converge, a bunch of them converges on a single mitral cell in the olfactory bulb. Then we have the cyclical circuits, reverberating circuits that control repetitive activities such as breathing. And parallel after discharge, where independent signals go through multiple pathways, these circuits are usually involved in higher mental processes. So what do you need to know about the circles? You should be able to match the verbal description of a circuit and its name and know general activities that are controlled by the circuits. Okay? Now, we're done with this. We're going to take like 5-10 minutes break and we'll talk about brain anatomy.